Hello, everyone. Thank you very much for joining us today for our first research seminar series ever that is entirely online. We are broadcasting from all over North America right now with Caitlin in Florida, actually. So there is some pretty cool advantages to doing it in this format. I'm sure a lot of you are all over the country as well. We should just get into it. So just a quick background for anyone who's not familiar with BMSC, we're a not-for-profit registered Canadian charity. We're a shared campus of the University of Alberta, British Columbia, Calgary, Victoria, and Simon Fraser University. And this seminar series is an ongoing thing that we've been doing for years. We usually host it on site in Bamfield, British Columbia, for students, researchers, and staff especially as well, to learn about all the exciting things happening in marine science. So if you like what we're doing and want to see more, please consider making a donation at bamfieldmsc.com. That's our homepage. We'll bring you right to a donate page. So I'll also just go over the logistics of this talk. Caitlin's going to give us a talk, and then at the end, we're going to have a time for a Q&A. So you're free to type questions in the Q&A section of your Zoom webinar page, and at the end, and click ask when you're finished with that. And at the end, Caitlin will go through some of those. We'll also be able to ask any questions that we receive through YouTube. I'll just ask those directly to Caitlin. Oops. I'll just move us away from here. So Dr. Gallagher, I had the personal pleasure of getting to know her last year when she came in 2019 to join the BMSC as a postdoctoral fellow. There she's been examining fluid dynamics of salmon and whale lice to calculate the drag of these animals and understand how they might mitigate these forces. So sadly, Caitlin's leaving us very soon to actually joined the faculty at Christian Brothers University as an assistant professor of biology. But today we're going to hear about her studies on the evolution and adaptations of what she looked at in her PhD, as well as a little bit of what she's looking at in her postdoc now. So she was at the University of Connecticut and studied the morphological and molecular evolution of the tapeworms of the pelagic thresher sharks. So really excited to hear about that. And I'm just going to stop my screen share and spotlight Caitlin here. I think you might still be muted, but if I do that.
I believe I'm unmuted now. Can you hear me? Okay. All right. Thank you, Luke, so much for that introduction. And I am going to move over to sharing my screen. Right. Okay. So again, thank you so much for that introduction, Luke. And I want to thank all of you for joining us online today. I'm sorry I can't see you all in person, but I'm really excited to still be able to talk to you about the research that I have been doing over the past few years. As Luke said, I'm currently the Bamfield Postdoctoral Fellow, and I am an evolutionary parasitologist. So basically that means that my work has focused on the evolution and diversification of parasitic species. However, before I actually get into my work itself, I wanted to go over a few parasitological terms and concepts just to ensure that we're all on the same page throughout the talk. So what I would like to start with is actually the definition of, if my slide wants to go, there we go, the definition of parasitism. So what is the definition of parasitism? Well, this is an interaction between two separate species in which the larger of the two species is the host and the smaller of the two species is the parasite. Now, this is a long-term interaction that is going to occur over at least usually several hours, if not days, weeks, months, and sometimes actually for over several years. And in this, we are seeing the parasite benefit at the cost of the host. And typically there's a biochemical or nutritional basis with this. Often we're seeing the parasite basically feeding on the host. Now with parasites, we can actually put them into two broad groups based on where they're found on or within the host itself. So the first of those groups is the endoparasites. Those are going to be the parasites that are found inside the body, in the organs, the blood, the muscles, and so on. Our second group are the ectoparasites, and those are going to be found on the surface of the body. So we'll be seeing them on the skin, fur, gills, and so on. And my research looks at both of these groups of parasites. I'm going to start by talking to you today about some endoparasites, and then I'll end the talk today with talking about a few ectoparasites. So now that we've talked about parasites a little bit, I want to start thinking a little bit more about the hosts. So what animal can be a host organism? Well, pretty much any animal on the planet that you can think of, vertebrate or invertebrate, is likely host to at least one species of parasite. And honestly, most animals on the planet are host to a multitude of different species of parasites. And because of this, it's often thought that parasitism is actually one of the most efficient lifestyle strategies that we see on our planet. And this is evidenced by the fact that parasitism has evolved over 60 different times in multicellular, so metazoan animals alone. And 40% of the world's biodiversity is thought to consist of parasitic species. So there is so many parasites out there. However, many of them are severely understudied. There's a lot we have yet to learn about even just their basic biology, let alone their ecology and evolution. So my research is trying to fill a few of those knowledge gaps. So now let's turn to actually look at some of the goals of my actual research program. So I have three overall goals of my research program. The first being to discover and describe novel parasitic species. As I've said, there are pretty much every animal on the planet serves as a host. However, there's many, many animals out there that have yet to be examined for their parasites. So we think there's many parasites left to be found and to be described. My second goal is to actually look at how parasites evolve. And I am interested in kind of two aspects of this. The first is the evolution of parasitism itself. How do you get a group of animals that go from a free living lifestyle to a parasitic lifestyle? Now that's of course a very large question, and I'm still working on building a system to look at that. So I'm more focused on this second part right now, and that is the evolution of species that are closely related. Particularly, I'm really interested in the diversification of species and the evolution of novel morphologies among closely related species. And the third research goal that I have is to understand how parasites adapt to what's often a rather harsh environment. And keep in mind, for a parasite, its environment is its host. So it's living on or inside an animal. This animal really doesn't want those parasites there. So it's a hostile environment. They're having to fight the immune system. So I really want to better understand how parasites have been able to overcome these really difficult and bizarre challenges of this lifestyle. So for my talk today, I'm going to start with these first two goals. And I started looking at these two goals when I was doing my PhD at the University of Connecticut. And for both of these goals, I use elasmobranchs as my host system. And elasmobranchs is the group of animals that contains sharks, stingrays, and skates. So why did I choose this as my host system? Well, honestly, because they're fantastic hosts. So sharks and rays host over animals from 7 different phyla that are parasitic, including species that range from leeches, parasitic copepods, lice, tapeworms, flukes, and so on. So they have a bunch of different types of parasites that they host. And these parasites are found all throughout the body of the shark. They're found everywhere from the eyes, brain, gills, heart, liver, digestive tract, skin, muscles, and even in the anus of these animals. So pretty much anywhere that you look in a shark, you could potentially find a parasite. And in fact, our current estimates of the number of different parasite species that sharks and rays host overall is around 2,000 different species. However, we believe this is actually a huge underestimate because only about 40 to maybe 50% of different shark and ray species have actually been examined for parasites. So that leaves potentially hundreds of different species that still have yet to be looked at for new parasites. And a good example of this comes from some of the work that I've done recently. In 2017, I had the chance to actually travel over to Taiwan with my PhD supervisor at the time, Dr. Janine Kyra, and our colleagues in Dr. Kirsten Jensen's lab. And we visited the fish markets of Taiwan. And basically what we did is we examined as many shark and stingray specimens as possible for parasites. And the one that I'm going to highlight here is this tiny little shark. This is the small-eyed pygmy shark, Squaleolus alii. And this is a tiny little shark. It can fit in the palm of your hands. And we found this basically in the bycatch bins from deep water trawls. So this isn't a species that's targeted by the fisheries, but it gets picked up by them. And this really, the species hadn't been looked at for parasites before. So we had the opportunity to take a look. And when we looked in the digestive tract of this shark, we discovered 2 tapeworms that were brand new to science. So completely undescribed. And we were lucky enough that we got just enough specimens of this tapeworm here to actually describe it. So me and Dr. Kyra described this tapeworm and named it Acanthobothrium catherinii. This species description I'm hoping will be coming out in the Journal of Parasitology in the next month or so. And this species was actually named in honor of my mother. So looking at just this one tiny shark, we're already finding 2 new species of tapeworm. So imagine if we examine those other 100 species of sharks that haven't been examined yet, that number 2,000 is going to go way up. There's so many more parasites that we have yet to discover in this group of host organisms. Now, I'm also fascinated by the evolution of parasites within the elasmobranchs. And particularly, I'm fascinated by the evolution of tapeworms. They're one of my favorite groups of parasites. So for my PhD, I focused in on one shark species and one group of tapeworms to really get to better understand their evolution. And the groups I chose to focus in on are the tapeworms of the pelagic thresher shark. So the pelagic thresher shark is a big shark, gets over about 10 feet long. It has this gorgeous long tail that it uses basically to stun its prey fish that it feeds on. And it's host to a small group of tapeworms that are closely related, as I'll show you in a second. And this was really quite a fascinating system to work on for my PhD, because the more I looked at it, the more I realized I was looking at this like mini family drama that I was trying to understand better. And I really am excited to tell you the story about this. But first, before I get into those details, I want to first do a little bit of a Tapeworms 101, just to define some of the terms that I'm going to be using throughout the rest of this talk. So tapeworms of sharks are always found in the intestines of the shark. They basically attach to the intestinal wall, as you can see here, and then they just kind of hang out there and let the shark basically digest down the food, and they absorb the nutrients from that food across their tegument, which is basically their skin. Now let's take a up-close look at a drawing of one of these tapeworms. So this is one of the tapeworm species I'll be telling you about today, and this is a drawing of it. And for tapeworms, they have 2 basic regions to their body. The first region up here is referred to as the scolex, and basically this is the attachment organ for this animal. This is how it actually attaches to the side of the intestines. And we see a lot of diversity and form of this attachment structure. So some have suckers, some have hooks, some have muscular pads. But for the species that we're going to be looking at today, they have a sucker at the top, and then they have these flaps that they use to help basically hold onto the sides of the intestine. Now, this is a pretty small region of the worm, right? The rest of the worm we refer to as the strobila. And basically, it is a chain of packages, and each of these packages is called a proglottid. And what the proglottid is just literally a package of reproductive organs. tapeworms are hermaphroditic, so they have both male and female reproductive organs. So basically these little packages are just cranking out as many eggs as they can. So this entire portion of the body is pretty much solely dedicated to reproduction. Okay, so now that I've covered some of the terms and showed you the basic body plan of a tapeworm, let's switch back over to thinking about the tapeworms of the pelagic thresher shark. So for quite some time now, it's been known that the pelagic thresher shark is host to 3 species of tapeworm that all belong in the genus Lidobothrium. So these are all pretty closely related. However, it was discovered a few years back by my PhD supervisor and her colleagues when they examined some of the intestines of this shark from Mexico and Taiwan, that there's a 4th worm that lives in the intestines that I just put up here. However, this is, as you can probably see, an extremely weird looking worm. And when my colleagues found this, they had no idea what they were looking at. So they were out in the field, had no idea what they found, basically put this in vials, brought it back to the lab so that they could further look at it, because honestly, it's incredibly strange. It doesn't really look like any of the worms that you would expect to find in the intestines of a shark. It doesn't really look like a tapeworm, doesn't really look like a roundworm, and it doesn't really look like a thorny-headed worm. So what in the world is it? Well, when they examined it closely using a light microscope, they found that this back region here was actually a tiny chain of proglottids. And if you remember what I said before, proglottids are part of the tapeworm body plan. So this is actually a tapeworm. It's the weirdest tapeworm I or anyone I think else has ever seen, but it's a tapeworm. So that means maybe this region up here is a scolex, but they weren't really sure. So they now knew they had a tapeworm, and they were certain that this is a new species. So the next step would be to describe this new species. But to do that, they need to know what group it belongs to, because typically you send the specimens to the expert that works with that group so they can do the description. So their next question was to figure out, okay, what group of tapeworm does this belong to? Well, there are 19 orders of tapeworms. There's a lot of tapeworms, 9 of which occur in elasmobranchs. So basically, there's 9 possibilities. The first best guess is would be the fact that it belongs to the same group of the other tapeworms that occur in the pelagic thresher shark, and those are the Lidobothridians. However, if you just compare the morphology, it seems highly unlikely. So at first, this was kind of discounted. It couldn't really possibly be one of the Lidobothridians. It didn't look anything like it. It doesn't share any characteristics. So what my colleagues did was they sent specimens of this worm to tapeworm experts all across the world, basically, so that they could take a look at it and decide, does this tapeworm belong in their group? However, after traveling around the world, it actually ended up getting back to the States and back into the hands of my PhD supervisor, Dr. Kyra, and basically there was no answer in sight. The morphology did not allow them to put it in any of the known tapeworm groups. It didn't look like anything we've ever seen before. So what do you do at this point? Well, they decided to look at the DNA, of course. So they did a DNA extraction and they ran a phylogenetic tree using the barcoding gene for tapeworms, which is the ribosomal 28S gene. And what I'm about to show you is a subset of the tree that they got back from that analysis. Now, I'll note that they had nicknamed this worm mushroom, as you can probably guess from its morphology. That's where it's got its nickname. And that's the name that you'll see appear on the tree for this worm. So our mushroom type tapeworm places robustly within the genus Lidobothrium. I'll remind you that that's the tapeworms that occur in the pelagic thresher shark. So a surprising result, but we now know what group it belongs to. It's a Lidobothridian. So my colleagues at that point were able to actually describe this tapeworm, and they gave it the name Lidobothrium enigmaticum. And this is the name I'll be using to refer to this tapeworm throughout the rest of this talk. So now we know that the pelagic thresher shark has 4 species of Lidobothrium tapeworm living within its intestines. However, this is such a weird result. And I'm going to show you why now, because honestly, this worm is nothing like its closest relatives. So what I'm going to do now is actually show you all of these odd differences between our weird species Enigmaticum and its closest relative Lidobothrium dalei. So the first thing I want to compare is the scolex. So remember, that's the attachment organ. For our typical tapeworm, we have that sucker and those flaps, right? Well, clearly that's lacking in Enigmaticum. We don't see those anywhere, right? And in fact, when they described the species, they found that this whole region here is the scolex. So the scolex on this worm is massive. And that means that the rest of this body plan, here on the typical tapeworm and here on Enigmaticum, that is the reproducing region, that is the strobila. So basically what we're seeing is a complete reversal of the typical proportions that we see in a tapeworm. So this is highly unusual. The other thing that's different is the size of this worm. So I've been showing you these worms at the same size just so you could see all the structures clearly. Well, this is actually the true size differences that we see between Enigmaticum and dalei. Enigmaticum is about 4 times larger than any of the typical tapeworms that we find in the thresher shark. So much bigger as well. The other thing that was discovered when Dr. Kyra and her colleagues were describing this worm was they had closely examined this scolex region here, so this attachment area, and they had discovered that there are these 4 weird cell types that occur in this region that have never been seen before in any other tapeworm. So another oddity, and the last oddity that I will show you about this unusual tapeworm is the way in which it actually interacts with its host. So all the other tapeworms in the thresher shark do not elicit any type of immune response. However, Enigmaticum elicits a severe inflammatory host response. So what you're looking at here, this is the posterior end of the worm, and then all of this here is host tissue that has swollen up and around the front end of the worm. And if I show you a cross section through here, this is the worm in the center, and all of this is inflamed host tissue. So it's causing this severe inflammatory host response that we haven't seen in any other Lidobothridian tapeworm. So all of these oddities and the unusual body plan of this worm was reported in a paper in 2014. And this is when I actually entered the picture and started working on this project. I started by reading through this paper, getting all the information we had on this worm, and forming the 3 questions that I really wanted to answer with my PhD. And those 3 questions were, first of all, what in the world are those odd cells that are in the scolex of Lidobothrium enigmaticum? They were only examined with light microscopy by my colleagues, so I wanted to get a closer look at them and see if I could get a better understanding of where they were and what maybe their potential function was. My second question was, how does enigmaticum cause that inflammatory host response? And the secondary question to that, of course, is why does it cause that host response? And then the final question I had was, how did this whole new body plan even evolve? So I really wanted to look at what molecular changes may have happened that allowed for this evolution of this new body plan to occur. So let's begin by looking at my first question. So in order to look at this, I really wanted to get a really close-up look at the internal anatomy of this tapeworm. So what I did is I took specimens of this tapeworm, I serially sectioned it, and examined all of those sections using a transmission electron microscope. So this is a high-powered scope that lets me look at basically the cellular level of this worm, right? And I was looking at images like this, where this is one large cell with the nucleus in the center. And then images like this, where we're seeing about 5 to 6 different cells here, and we're seeing the nuclei here. So what I did is basically looked at all the cells that I could find in this region of the worm, and I characterized the different cell types that I found using characteristics such as cell size, nuclei size, cytoplasm to nucleus ratio, and so on. And I found that there weren't just 4 new cell types in this worm. There are actually 11. So there are 11 unique cell types. And again, we've never seen these in any other tapeworm so far. So what I'm going to show you now are the electron micrographs of each of those 11 cell types. So I've given each of them these letter designations. And I know that there's a lot to look at here, but the one important thing that I want to point out to you right now is the fact that every single one of these cells have some type of vesicle in them that seems to contain products that were produced by the cell. Furthermore, 3 of those also contain these inclusions that contain a second type of product that seem to be produced by these cells. So I have 11 cell types within the scolex, all of which are producing some type of product, some of which are producing potentially 2 types of product. And now what I want to show you is actually where you find each of these 11 cell types in the scolex and how they're actually associated with one another. So this is the schematic I put together. Here is the key to all the different cell types that I just showed you. And you can see that these cell types are all occurring throughout the center region of the scolex here. And as you can see, they're really tightly associated with one another. And they're even forming concentric rings at some points in the scolex. So this is a pretty complex system of cells that we have found here. Now what I want to do is turn to my second question, because I think the answer to this question, how it causes this inflammatory host response, is tied to those 11 cell types that I just showed you. Because if we look at the region of the worm that's encapsulated by this plug of inflamed host mucosa, we find that it's this entire region that contains those 11 new cell types. So my hypothesis is that the products that are being produced by these cell types may be helping to elicit this inflammatory host response. In order for that to be true, of course, though, that means that some of these products are being released to interact with that host mucosa. So I had to look for a way for that to be happening. Well, I went back to the 2014 paper where my colleagues and collaborators actually described this worm and found that they said that there was an aperture at the very top of the scolex of this worm. So that's basically this region here. So I'm thinking that some of these secretions are probably being released through this aperture, but I really needed to confirm this. So I went back to all the sections I took and looked at the sections I took from this region, and I was able to find some of the aperture. So this is the aperture here. And you're seeing this gray product in the center of the aperture. And I believe this is the secretory product from some of those vesicles and inclusions that I showed you earlier. So it does appear that this worm is releasing some secretions that can come into contact with the host mucosa. So I do believe that at least some of the products that are being produced by these cell types are causing this inflammatory host response. Of course, we need to do a little bit more research to confirm that. Now, the natural follow-up question to that is why? And again, this is just a hypothesis, but honestly, I believe that this is the tapeworm's attachment strategy. Because if you look at the scolex proper here, you don't see hooks, you don't see suckers. There's no way for this tapeworm to hold on to the intestinal wall of its host. But having this host mucosa inflame around it is actually a really good way to make sure that tapeworm doesn't go anywhere. Because when I go in to remove these worms from the intestines, when I remove the typical tapeworm species that just have the sucker, I can tap them and they kind of just come off. They're really easy to remove. When I go to remove enigmaticum, it's an ordeal. I have to cut them out, and it's really hard to do that without damaging the tapeworm specimen. So this is a really good way of making sure that that tapeworm's not going to move anywhere. So I believe I've addressed those first 2 questions. And now for the rest of my PhD, I wanted to focus on this last question to really start to understand what changes occurred on the molecular level that allowed for that novel body plan that we're seeing in enigmaticum. And in order to look at that, I needed to look at, one, the genomes of these animals. So the genome is all the DNA within an organism. And I wanted to look at the genome of enigmaticum, my weird species, and compare it to 2 of the normal tapeworms for differences. So I was able to do DNA extractions, put together the genome, and then I could look for differences such as deletions, insertions, reversals, or even potentially duplications with either of genes or potentially even whole genomes in order to look to see if any of those types of changes could underlie that difference in body plan that we're seeing. The other thing that I wanted to look at was the gene expression. And to look at that, I needed to look at the transcriptomes. So with the transcriptomes, I could basically look at what genes are being utilized at the life stage that I was looking at, which is the adults. So which genes are basically being turned on and used. So in my schematic here, green means it's being used, the gray means it's not. And in this example here, one of the differences I'm seeing is, say, gene 5 is being turned off in the weird species, while it is being used in these other species. So I'm looking for some differences like this in my transcriptomes, but also, of course, an overall expression levels for the ones that are turned on, any differences in the number of gene copies that are being made for a gene. And that's shown here. Each of these blue lines is a gene copy. So I can look for differences such as, let's look at gene 1, where in the weird species, there's only a few copies being made. And in the normal one, we're seeing a lot more copies being made. Or I could look for I might see differences, that we see in gene 6, where lots of copies are being made in the weird species and only a few in the normal one. So these are some of the differences I was looking for when I looked at my transcriptomes. So in order to do this, I collected specimens actually while I was in Taiwan in 2017 of all my different species. I digested them down, and I was able to do extractions that gave me DNA for my genomes and RNA for my transcriptomes. Now, I actually did all the library prep, the sequencing, the assembly, and annotation myself. However, I don't have time to get into those details today. If you have any questions about the pipelines and methods I use for that, I would be happy to talk with you at either the end or at any other time about that. But after doing all of this, I did end up with both genomes and transcriptomes that allowed me to go forward with actually looking for the differences that might explain this novel body plan. So first, I got 3 genomes. My genome for my odd species Enigmaticum, and for 2 of my normal species, one of which was that close relative Dalei. And I was able to start looking for differences between these. And honestly, the easiest difference to look at right off the bat was just the genome size. And what I found is that the genome size for Enigmaticum, 300 megabases, is extremely similar to that of Dalei, 301 megabases. So very simply put, what does that tell us? Well, basically, I'm not seeing anything like a whole genome duplication, which in other systems has been found to lead to morphology changes. We're not seeing that in this system. The genome sizes are too similar, and we think that these 2 species speciated very, very recently. So I don't think this contributed to the novel body plan of Enigmaticum. Another thing that I looked at was potential expansions within gene families in an Enigmaticum. And what I mean by that is genes are often put into families, and in those families, all those genes will serve a particular function. So we could say that this was a family in which these genes contribute to, say, immune response. And I was looking for instances in my weird species where maybe I'm seeing an increase in the number of genes that are within that family, say go from 3 genes to 6. So I did this using what's called a CAFE analysis. And I found that there have been several gene family expansions within my weird species. One expansion that I've seen is that there are expansions in gene families that are associated with anatomical structure. And that makes sense because we're seeing a completely different morphology and internal anatomy of our new species. I'm seeing expansions of gene families that have to do with secretory products. Again, that makes sense because Enigmaticum has those secretory products in each of those 11 new cell types. And then also in gene families that are associated with immune response. Again, this makes sense to me because Enigmaticum causes that severe inflammatory host response while the normal species do not. The other thing that I looked at was the gene expression, so the number of gene copies that were made. And that led me to be looking at figures like this. So I know this is a little bit overwhelming potentially, but basically what this is that each of these rows is a gene, and the coloration is telling you how many gene copies are being made. So if it's red, lots and lots of gene copies are being made. If it's blue, basically fewer gene copies are being made. And I wanted to look for differences in Enigmaticum, that's over here, compared to my 2 normal species. And I see some regions like here and here where I'm seeing some differences. However, when I run my statistical analysis, honestly, most of these differences aren't actually significant. There's only a few things that come out as significant, and those are that there's higher expression, meaning a higher number of gene copies made for genes associated with anatomical structure. So again, that would make sense in my mind. We're seeing a completely different anatomy and morphology in this worm compared to this worm. But we're also seeing lower expression, so fewer gene copies made of immune response genes within the weird species. So that tells us that enigmaticum is doing something different when it comes to immune response than the two normal species are. And that, again, makes sense to me because we're seeing a different response to enigmaticum with this swelling than we are with the normal tapeworms. So this is work that is still ongoing. And I am working on redoing a few analyses and improving some of my annotations to get a better answer to some of these questions. But I'm hoping that within the next few months, I'll have potentially a few more leads on the molecular changes that are actually occurring and be able to publish them within the next few months. So overall, what I've found is that there are 11 unique new cell types within the scolex of enigmaticum, all of which are producing some type of secretory product. And I believe that potentially that some of those products may be helping to elicit that inflammatory host response that we see associated with enigmaticum, and potentially that could be helping with attachment of the worm. As I just said, I'm still looking into the potential molecular changes that underlie the novel body plan of enigmaticum, but what I've found so far is that there have been some gene family expansions and some gene expression differences, but I'm hoping to have more to look at and a more solid answer to this question within the next few months. So I hope now that I've gone through this, you can see what remarkable host organisms elasmobranchs are to look at. They have an amazing and massive diversity of different parasites. And then the evolution and ecology of these parasites, as we look closer at them, is really fascinating, but we have a lot more work to do, as I said, in this system. So now that I've talked about this, I hope that the next time you hear about sharks or hear the word sharks, that instead of this particular image coming to your head, that maybe something more like this comes to your head as well, and you marvel at them as being these magnificent creatures that are home to many other species. So now I want to transition from my first two research goals over to my final research goal. So as I said, I've always been interested in how parasites are able to deal with their environment, but I didn't have the chance to actually look into this question until I got to starting my postdoc at Bamfield. So this work I just started this past year, and I'm going to share a little bit of the system that I'm working on and a few very, very preliminary results, but I'll hopefully be able to update you more in the future about what we've been doing. So at Bamfield, I started working on this project, and I have been working with Dr. Chris Cameron, who's my postdoctoral supervisor. And basically for this project, we have been combining his expertise with fluid dynamics with my expertise in parasites. So what we've decided to do is really look into the fluid dynamics of ectoparasites in two different systems to really kind of understand better from the parasite's point of view what kind of stressors that they're dealing with while they're attaching to their host. And the first system that we decided to work on is one that's, I'm sure, near and dear to every Bamfielder's heart, and that is salmon. And as I'm sure everyone in Bamfield or on the West Coast is aware, the salmon are dealing with a huge problem with sea lice, and that is my parasite that I'll be studying in the system. So sea lice are parasitic copepods. They feed on blood and mucus, and they've been highly problematic, especially in the smolts, as I'm sure many of you know, because high infection levels can lead to death. And with this system, what I'm really interested in looking at is understanding how the water is moving over these parasites as they're attached to their host, and being able to calculate the drag that they're experiencing. And I think this is important because if we can calculate the drag that these individual parasites are actually experiencing, we can have a better and more holistic understanding of the stress that that's putting on its fish hosts. Now, the second system that we decided to look into is another charismatic system, the ectoparasites of whales. So whales are host to these little creatures known as whale lice. They are parasitic amphipods, and some whale species have one species of whale lice. Other whale species will host several different species of whale lice, but all of them will usually host hundreds to even thousands of these lice. So what you're seeing, this white clump all here and here, those are all whale lice attaching to this whale. And as you can see, looking at this whale lice specimen here, they've got really bizarre body morphology. So me and Chris are really intrigued about how does the water move over that, because that doesn't seem like a good body form to reduce drag, and to be able to better calculate the drag. And with this particular system, we're also really interested in understanding if they are helping to reduce that drag somewhat by the placement on the whale or the way that they tend to group up together. Okay. So to begin this project, I arrived in Bamfield in August of last year. And of course, the first thing I had to do was collect specimens. So I collected one species of salmon lice from fishermen in the Bamfield community, and a big thank you to all of the fishermen who allowed me to come onto their boats and pick lice off their fish. I'm pretty sure most of the fishermen in the community now know me as the Parasite Girl, a nickname I will say I absolutely embrace. And I also had some wonderful colleagues from California that agreed to send me whale lice specimen that I could use. So that was Dr. Christopher Callahan from the College of the Redwoods, and also the Cascadia Research Collective. Once I had all my parasites, I was able to then start heading into the lab. And what I was going to do is use a flume, and I did this both in Bamfield and Montreal, to look at how the water is actually moving over these species. So how did we set up this system? Well, if we zoom in here, you see that I attached our little specimens using modeling clay. Here we have a little whale lice. And what we would do is we have this laser beam coming down and forming a plane, and I seeded the water with these micro beads that when they float by the laser, they'll actually fluoresce. And what that does is it allows me to see how the water is actually moving up, over, and around our parasites. So it gave me images like this. So here's our parasite, and all of these are the fluorescing beads that you see here. And that gave us videos like this. So you can see these particles moving up and over our whale lice here. So I did this with one species of salmon lice, and I did it for 4 species of whale lice. One whale lice species was from a humpback, and the other 3 were from gray whales. Okay. So I was able to do all the videos I needed for this while I was in Montreal. And looking at these videos, I was able to get a rough idea of how I think the water is moving around these animals. And I'm going to show you some quick schematics of that now. So for the whale lice, there we go. Here is our little rough cartoon of a whale lice, the real specimen up here. And what I observed from the videos is it looks like the water's coming up, hitting the anterior end, going over, and we're starting to get some eddies once it gets to the posterior back of the lice. I also believe there might be some water flow underneath. And this would make sense because one of the whale lice species actually has its gills underneath in the ventral area, and all of the females of all 4 whale lice species have brood sacs under there where they keep their young. So it makes sense that they would have to keep the ventral region oxygenated. The water flow over the salmon lice, on the other hand, is a little bit more straightforward, where it goes up, over, and it seems to form eddies towards the posterior end of the organism. So after doing those videos, I have a rough idea of how I think the water's moving over them. But with this really odd and complex morphology of the whale lice, I'm not 100% certain that I might not be missing something in those videos. So the next step to do is really to actually model the water flow using computational fluid dynamics. And in order to do that, what we need is 3D models of my parasite species. So what we did is we took 1 species of salmon lice and 2 species of whale lice, and we sent them for CT scanning, and then had, with the help of our colleague in Oxford, Imran Rahman, he helped us stitch those scans together to get 3D models. So currently I have a 3D model put together for the whale lice, a rough one for the salmon lice. However, we had not been able to get the data yet back for the second lice species before all the universities shut down due to COVID. So we're currently waiting until things get at least semi back to normal so we can get the data for that and form our third 3D model. Once we have that, we'll be able to move on to our next steps, which is the computational modeling, where we'll actually model the flow of water over these in an application such as COMSOL. So I'm hoping maybe in a future summer, I'll be able to actually present those results to you as we get further on in this work. But I'm really excited to see what we find and see how well it matches up with what we've found in the videos we've taken already. So I would like to thank everyone who has helped me with my work, all my amazing collaborators, everyone who's taught me all the techniques I've worked on and provided me with specimens. Of course, thank you to all my funding sources. Thank you all so much for your attention. And I will take any questions at this time. So I think we have a few in the Q&A. Okay, so I see one question in the Q&A so far from M. Hi, M. Sorry if I missed this, but why are elasmobranchs so heavily parasitized compared to other groups? That's a really good question. To my knowledge, we don't really know why exactly they make such great hosts. It just seems like certain groups are better hosts than others for whatever reason, and elasmobranchs seem to be one of them. Maybe it's the fact that they can occur in, I'm guessing, many different niches, since you have some that are those top predators that are occurring at the top of the food chain, but you also have those that are mesopredators. So those can serve as final hosts themselves, but also as intermediate hosts. So maybe that's part of it, but we're not particularly certain why, in fact, that they're such good hosts. And it's something that came out of one of the big projects that my PhD supervisor was a PI on, where we were looking at the tapeworms are the vertebrate bowels of the earth. And one of the big things that popped out of it was like, wow, sharks are sharks and rays are really good hosts, but why more than others? We're not quite so sure. Hi, Chris. So Chris just asked, how does your mom feel about having a parasite named after her, and what's with the thresher shark tail? So my mom, I was actually a little nervous, to be honest, to tell her when I decided to name the parasite after her, because I know for taxonomists, we all know that's a big honor, but I wasn't sure how she would feel about it. But I was really, really relieved when she felt really honored. She knew how big of a deal it was for me. So I think it meant a lot to her. And she actually has a framed picture of one of the scolices for that tapeworm that I drew hanging on her kitchen wall. And for the second part of your question, the thresher shark tail, it's incredibly elongate because they actually kind of use it as a whip to stun their prey. So they'll go past fish, whip it around, smack them, stun them, turn around, and then eat them. So it's the way that basically they hunt. Okay. The next question comes from Daniel. Have the guts of other species of thresher sharks been investigated yet to see if they have relatives of Enigmaticum? Great question. So yes, there are Lidobothrium tapeworms in the big-eyed thresher shark, but for some reason not the common thresher shark. We're not sure why that is. And we think there potentially might be something similar to Enigmaticum, but we're not sure. Honestly, we need to look at more intestines. But there are definitely Lidobothrium tapeworms in it, but they are more of the normal type tapeworms. So if anyone knows anybody that has spiral intestines for the big-eyed thresher sharks that would need to be examined for tapeworms, I'd be happy to take a look. And let's see, the second part, it would be interesting to build phylogenies around that and see if it matches with the phylogeny of, I'm guessing, I can't see the rest of that, but I'm guessing the phylogeny of their host. And yes, I think that would be really interesting to look at. We currently don't have a phylogeny of all the Lidobothrium species yet. So that's something that we would absolutely love to do and see if it matches up. I would honestly say, though, that I wouldn't be surprised if it doesn't match up, though, because we're not seeing a lot of co-phylogeny between tapeworms and elasmobranch hosts so far from what I've seen the other elasmobranch tapeworm experts doing in my field. We had expected, I think, to see more co-phylogeny, but we're not seeing too much, actually. Okay. The next question is from Colin. What negative effects can tapeworms have on their shark hosts? So mostly it's just that they're taking up nutrients from their host. So they're stealing away some of those nutrients. However, it honestly doesn't seem to affect them that much, really at all. When I open up a shark, I can never really tell a parasitized from an unparasitized ones. And honestly, most of, at least the thresher shark valves that I've looked at, they're all pretty heavily parasitized with Lidobothrium tapeworms. And all the sharks seem fine. They seem happy. Well, not happy. They're unfortunately deceased when I look at them, but healthy is the word I was looking for. They all seem healthy. They don't seem like they're underweight or anything like that. So they take nutrients, but not enough that it seems to really harm the host. Joanne, are there other parasites that cause the immune response you're seeing in the sharks, maybe in other animals? Do those parasites also have cells that are exhibiting the secretion packets in enigmaticum? Great question. So there's only a few other species of tapeworm that cause some type of inflammatory reaction. But the odd thing is in my system, it's one parasite that causes that huge inflammation. In the other tapeworms that do this, it's often a bunch of tapeworms attached at one point that causes more of a buildup. So it's a little bit different where it seems like there has to be several tapeworms that cause it. But I have looked at the literature and looked at the work that's been done on their internal anatomy, and it seems like some of them may have like secretion glands, but nothing that looks like that what we see in enigmaticum. Nothing close. Kelsey, I wouldn't mind hearing more about the life cycle of the thresher shark tapeworms. How are they transmitted to their shark hosts? That is a great question. We know the rough life cycle for them, but we actually don't know all the specifics of the Lidobothrium life cycle. And honestly, for many of the described tapeworms out there, we don't know their full life cycles. They are trophically transmitted parasites, so they move through the food chain. So it's thought that the eggs are out in the open environment. They're probably eaten by some type of small planktonic creature, maybe a copepod, which is then eaten by maybe something larger, either a larger invertebrate or a small fish. That's likely eaten by a bigger fish and then eaten by one of the sharks, is what we think the chain is likely is. But like I said, we actually don't know the intermediate hosts for any Lidobothrium tapeworms at this time. It's something that would be really fascinating to look into, but there's a lot of possibilities because tapeworms tend to be less specific for their hosts at the intermediate stage. They can infect more species versus their final host stage. Many of the elasmobranch species will only actually infect one species of shark or ray. Any other questions? I see no more in the Q&A.
Stacy actually has a question here for you if we wanted to maybe wrap it up with one last question.
Yeah, great.
Yeah, I'll let her ask it here.
Hey, Caitlin, I loved your talk so much.
Thank you.
I am just so curious about how odd this thresher shark tapeworm is. And do you think that that's indicative of like a relatively long evolutionary history with this host? Or is it perhaps kind of like a novel, like host jumping scenario where it's, you know,
Yeah.
it doesn't look like any of the other parasites that the thresher shark has because it's new?
So we don't know for certain, I will say, but my belief is that this is a long evolutionary relationship, because all of the Lidobothrium tapeworms within the pelagic thresher shark are each other's closest relative in the tree that we have so far. I should say that they could potentially change as we add in the few other species that we haven't sequenced yet, but so far they're a monophyletic clade that all occur within the thresher shark, which indicates to me that this, I think maybe potentially it's a long evolutionary history and not a host hop. So I think it's a recent speciation within the host.
Awesome. Thank you.
Oh, can I address this last one from Florian?
Absolutely.
Hi, Florian. Glad you could make it. Can you talk about, for the ectoparasite work, whether you're considering how the water flow would vary over the different parts of the fish or whale body? Yes, that's definitely something that we would be really interested in doing at some point. Currently, we are just looking, I'm just doing the flow over the flat surface right now just to kind of get the baseline. But I think we would really like to take it to that point of in the future where we're looking how the different parts of the body potentially will affect that. But just to start off, I wanted to start all kind of at a flat zero to get a rough idea overall and then move on to that.
Well, thank you so much, Caitlin. That was incredible. We were also happy to hear about something non-COVID research related for once. I think you only mentioned COVID once in the talk, so it was nice to have a break. And I encourage anyone to reach out to Caitlin with additional questions. You can always contact us for her email address, but I believe it's posted on the Bamfield MSC website as well. Great to see you all here. I'm just going to, I guess, screen share one more time and do a little plug for next week's seminar. So we have a big thank you to Caitlin once again, and we can't wait to hear more results next summer when you've had some results come in based on everything hopefully opening up again. Next week, we're really excited to hear from Dr. Isabelle Cote, who has been an instructor at BMSC for years and will be presenting actually on some of her longer term data sets collected during subtidal science, formerly scientific diving. Her talk is titled Ecological Shifts in Marine Community Following Extreme Disturbances. I'm not sure if it's her class that's doing the disturbing or if they're going to check maybe where the Alta has been dredging. If you're interested in seeing a live dredge that's now posted on YouTube, please go to our YouTube page and check out part 1 or part 2 of that. Maybe you can see the disturbance pre the ecological shift in marine communities. So please go and sign up from the Zoom link that will be on the homepage, bamfieldmsc.com. And I'm not sure if you can do this in a webinar, but feel free to use that applause button to give Caitlin a big applause. And otherwise, Caitlin, I'll catch up with you later just to talk about how great it was. Okay, thanks everyone. I think we have, thank you very much from Barb Beasley, and in the chat, lots of thanks. So yeah, we miss you here and we look forward to seeing you soon.
Thanks from YouTube.
YouTube is thanking you as well. Okay, goodbye everyone.